So, hello everyone, and welcome to this SONA webinar series, which is organized by the Society of Neuroscientists in Africa in collaboration with Trends in Africa, the African Academy of Neurologists, as well as the International Parkinson's Disease Genomic Consortium Africa. I'm Karin Gemeni, a research fellow at the University of Würzburg, Germany, and I am very happy to host the webinar today. The purpose of this webinar series is to make neuroscience accessible to all so that we can get experts from a broad range of research focuses to inspire early career scientists and also perhaps increase mentorship opportunities. And today I am very excited to introduce an impactful and multidisciplinary and inspiring researcher, Dr. Ali Ertug, who is a neuroscientist, an inventor, and an artist, all that under one hat. He has actually been recently appointed director of a new Helmholtz Institute on Tissue Engineering and Regenerative Medicine in Munich. And before that, he was an independent group leader at the Institute for Stroke and Dementia at the LMU in Munich, Germany. Dr. Ertug actually started his undergraduate study at Bilkent University in Ankara, Turkey. And then after that, he joins Max Planck Institute for Neurobiology in Munich for his PhD and went out in the USA, San Francisco for his postdoctoral research. He's also known for invention of disco tissue transparency technology and deep learning-based analysis of large 3D images in biomedicine. So maybe he's going to touch on that today. We're really excited about that. Dr. Ali Ertug has received numerous prestigious awards and fellowships, including the Early Stage Research Fellowship from Marie Curie program of the European Union. This is a very competitive fellowship, as well as the ERA-NET grant. And his research is published in high-impact journals like Nature Medicine, Neurons, Journal of Neuroscience. His work is fascinating, and we're really happy to host him in this webinar today. So Dr. Ertug, you're very welcome. Thank you for being here. Before we start, I really want to remind all the participants that this webinar is meant to be interactive. We have a chat section where you can actually post your questions as the webinar goes on, and Dr. Ertug will answer all those at the end of his talk. So now I'm turning the stage to him. Thank you, and we're all here.
Cool. Thank you, Karina, for the nice introduction. And thank you all the organizers for inviting me to share our research with the society here and the people. So let's do a screen share. My view will be gone, but I think the presentation at this point is more important. So let's get this. All right, so I assume you see my screen. Let me know if you don't. So since the beginning of human history, we desire for a longer and healthier life. Even actually an eternal life, which was the theme of the epic story of Gilgamesh, which is actually considered the first known literacy of human being. Look for an eternal life. Well, however, the truth is was a bit far away from an eternal life. Actually, thousands of years, people lived in average about 40 years. However, with the advent of modern medicine, we started to live now, around 60, mid-1900s, and nowadays we can live around 75 to 80. Well, because we could tackle infectious diseases and deal with accidents with modern hospitals much better. I call this period biosciences where we stop the premature death. However, as you see, the curve is flattening. The main reason you see on the right-hand side now, we cannot tackle actually some very important diseases from ischemic heart disease, neurodegeneration, lung diseases, and cancer, killing all the people. Still, more or less, there is no treatment for any of this. Well, you know, while the curve is flattening, we know that in maybe some 100 years, 300-400 years, we will be able to tackle almost all of these diseases, and we will be again in an accelerated longevity. Well, then, you know, as a scientist, and I ask myself, why do we need to wait several hundred years? What can we do to accelerate biomedical discoveries that we can have the progress of this 300 years in the next 20, 30 years? So I call that such an acceleration, which is not there, but if it happens, I call it bioscience era 2. Actually, this is also my primary vision as a lab, that to push the sciences into era 2, that things are much more accelerated. But how can we do that? So many scholars, and also as discussed in these 2 books, Homo Deus from Yuval Harari, Dan Brown, Origin, many scientific scholars as well think that we can do by merging enabling technologies. So we can really accelerate sciences by combining technologies. And this has been actually the core of my research since I started my lab about 5.5 years ago. So let's focus on biology, what I want to tell you today. As you know, we are an organism of interconnected systems. From head to toe, we are wired with vessels, nerves, and different systems. Therefore, it's clear that a disease affecting one part of the body will affect the rest. However, in the lab, we rarely study diseases at whole body scale. Simply, we don't have the tools to do so. If you think about, for example, imaging tools, on the one hand side, we have MRI, fMRI, DTI, like imaging modalities, where you can see gross anatomy, but you don't see the cells and molecules. If you want to see the cells and molecules, you can do histology, serial EM. But then the problem is there, you cannot scale this up to whole organ, forget about the whole organism. The technologies that we have been doing combines these 2, so that we see gross anatomy at subcellular level in a time and cost efficient way. So those are so-called optical tissue clearing. So what is tissue clearing? As you know, potentially dead standard histology relies on sectioning, labeling, and looking at the sections, which is most of the time has been very good and useful. But if you want to study complicated 3D structure of the organs and organisms, like vessel networks and neuron networks, that is not sufficient. So to overcome this problem, we and others have been developing optical clearing methods to convert opaque biological tissue into transparent structure. You can think this as converting milk into water. Now you can see through and scan through to obtain cellular level information throughout intact biological specimens. When I started my lab, we carried all these efforts to whole mouse body scale. First, we published so-called UDisco technology in 2016. And about 3 years later, we published in 2019 the VDisco technology. So as you see on the left one, the green mouse, actually where we rendered the whole mouse transparent, but at that time, for the first time, we could also scan a mouse with a laser scanning microscope. But you see this is a mouse that is labeling the nerves. The nerves are labeled, but still we have a lot of background. Later on, we improved that by enhancing the signal, GFP signal here in the neurons about 100 times. And you can see now on the right-hand side, all the nerves of the intact mouse revealed through intact bones and skin. So VDisco technology truly renders the whole mouse transparent. After rendering the mouse transparent, we put them onto laser scanning light sheet microscopes and scan them end to end. As a neuroscientist, we apply this first to see nervous system of a whole mouse. You see here type 1 GFP line mouse. A subset of neurons are labeled with GFP. Those are in green, and you have muscles in red, and white shows the organs and the bones. Now I'm going to remove the muscles. You can see the nerves through the bones and organs. If I remove bones and organs, you see the nervous system of an intact adult mouse. Let's look at this more in 3D video. You can see again the same. When we remove now the muscles and bones, you have the nerves. Here you can focus on any nerve from the toes, how they come backwards from the neuromuscular junctions and all this network of the nerves throughout the limbs. They come together from the bundles going to the lower part of spinal cord. You can see the ganglia, sensory and motor ganglia throughout the spinal cord, and the nerves that are innervating internal organs and chest, and a lot of nerves that are innervating the upper forelimbs in these animals. So as you see, we have now this really huge amount of details in intact adult mouse to do subcellular level even investigations. So next, what we did is actually to induce a traumatic brain injury on the mouse, on the right somatosensory cortex, instead of looking at only the brain, we also look at throughout the body. And if you look at the shoulder blades here, you can see that now these nerves that are innervating shoulder blades are highly degenerated in TBI case. On the control animals where there's no lesion, you see a lot of nerves innervating these shoulder blades. So this data already shows that if we look at even brain injury, a disease that are affecting exclusively brain is not actually affecting in reality in the brain, but throughout the whole body. You see the nerve changes throughout the body. We have been applying this technology to investigate different biological phenomena. We were very interested in meningeal vessels, which were rediscovered recently by Jonathan Kepnes, Kari Alitella, and Michael Nedergaard. So using this our transparency technology, now we can actually look at this meningeal vessels in an intact setting where the skull is there, the brain is there, and you see these vessels in between. But not only the vessels, but you can see also these immune cells, macrophages, monocytes, and other immune cells throughout these meningeal vessels. So studying this intact transparent mouse, what we noticed actually that there were some short vascular connections between skull marrow and the meningeal surface of the brain. We call it SNCs. You can see here pointed with yellow. So basically, this is in mouse. When we did it in the human, we saw the same, the human postmortem human skull samples with the meninges. Similarly, they are connected. So this data clearly shows that there is a connection between skull marrow and the meningeal surface of the brain, suggesting that the skull actually can be used to diagnose and treat the brain diseases from a new perspective. Imagine the immune cells in the skull marrow after an injury getting into the brain, causing neuroinflammation or others. Now we have to tackle, we could maybe tackle the inflammation, not in the brain, but outside of the brain. I think this is really exciting to pursue. So while we have been doing all these transparency technologies, of course, we generate thousands of images, terabytes, even hundreds of terabytes of data, and you cannot analyze them manually. To be able to analyze them, we have been using deep learning, machine learning part, where it can perform unbiased, high performance, and high speed data analysis. So I will give 3 examples. First is VESUP technology for vasculature, DeepMAC for cancer metastasis and drug targeting, and last additional technology for human organ mapping. In VESUP technology, we developed first a labeling method where we could label whole brain vasculature for mouse. combined with large vessels and tiny vessels down to capillaries. And then Johannes developed algorithms, deep learning algorithms, to analyze all this vasculature at the microcapillary level. You can see now this is in segmented data output from the WhatsApp. Here is the cerebellum, all the large data vessels on the cortex, but also if you dive in, you see throughout all these capillaries in the mouse brain. And then all this data is quantified and registered in Brain Atlas to get information readout in 1,200 plus brain regions. So we are using, of course, now this technology to analyze brain vasculature in different diseases. For us, an important application of technology has been also cancer metastasis, because many people, they die from the metastasis of the cancer, not really primary tumor. What happens is hundreds and thousands of cells, cancer cells splitting from primary tumor, they get into lymph vessels or blood vessels. The majority of them are eliminated by immune cells. Some escape this surveillance, exits the vasculature and hides in the different tissue regions, and then can form the large metastasis. And before our technology, the standard way of imaging this tumor bioluminescence, MRI, computer tomography, and others, they would give you all similar imaging. To be able to see something, you should have thousands of cells together minimum. So you're nowhere close to seeing there's a few cells that may form cancer, cancer stem cells, and in distal organs may form the large metastasis. We are really far away from that with these technologies. And we said we could solve this problem using now our technologies. We developed so-called DeepMak technology, where we transplanted the tumors into the mouse, memory carcinoma. After about 10 weeks, of course, metastasized, we made the mouse transparent and we look at throughout the body, and we image cancer metastasis at single cell level, plus drug targeting. Here also we have a therapeutic drug that is developed to target these cancer cells, which are labeled with another fluorophore, and we can image both of them. Using the technology, we map cancer cells and metastases throughout the body at very high details in 3D, quantify their size and their frequency. You can see here, for example, on the upper graph, there's about 120 single cancer cell, about 100 double cell size, and about 100 triple cell size, and so forth. When we perform all this technology, what we notice, about 60% of cancer metastases in the mouse body, they had the size of 75 cells and smaller. Actually, 75 cell here, I put it there because this was the best of any other technology can do. You can see even then they missed 62%, more or less 2/3 of them metastases in the mouse body. So you can imagine how accurate it would be to study cancer metastasis with other technologies. And we could also, of course, monitor the therapeutic antibody. Here, 6A10 antibody developed to target this tumor. You can see it's targeting in partial and in sometimes full. And using this technology, now we did it in whole mouse. I will show you here's a comparative image bioluminescence. There are about 4 tumors detected. In the red rectangle where you see the lungs, there is nothing visible. But after our technology, you can see all these hundreds of tiny micrometastases throughout the lungs of this animal. So let's look at this in 3D. Now we will fly through. You see the primary tumor close to the leg, the large one, and then there will be hundreds and a lot of micrometastases in the skin, in the bone, in different organs. And a lot of them were actually in the lungs in these animals, down to really few cells. You can see here in the bronchi and some collection of them. In addition, we could track the therapeutic antibody. Now, when the tumors are targeted with the drug, you see them, they are in white. And then the drug is not finding the tumor, they are in magenta. Here, you can see already many of the tumors are not targeted with antibody-based drug, which is supposed to be targeting cancer metastasis throughout. But then we notice about 20 to 30% of the metastasis still, they're not targeted with an antibody-based therapeutic. So at this point, I think this technology tells a lot by at the preclinical stage, where you can see the quality of antibody-based drugs at really cell level and say, here, this antibody potential is not ready to push to clinical trials because it's missing already 20%. Imagine to be able to treat the tumor metastasis, cancer metastasis, we would really need more or less 100% targeting. A few cancer cells that are left might be sufficient to kill person. So how can we translate all these technologies into human organs, human tissues? When we started, actually, things were not working as great. And then there were some problems. Sean, a chemist in the lab, solved these problems. So what was the problem? So to be able to permeabilize a tissue in general for antibody labeling in all technologies that we develop, clearing technologies, standard detergents are SCS and Triton. You see they have this mycelis size, which is relatively large, and they got stuck on the surface of tough, sturdy human tissue. They don't travel deep. And we did some screening and identified chops in new detergent with small mycelis size, which could travel throughout this tough human tissue and make the pores and then allow labeling and clearing. Using this technology, we rendered the whole human eye transparent. You can see it's fully rendered transparent. Then after light sheet microscopy scan, we can see the cellular details, high resolution details of intact human eye, iris, retina, and the behind the nerves and immune cells around. So not only, autofluorescence or dye, but we could even do antibody labeling in large human tissue here, more than 1 centimeter size, thickness of a human brain cortex labeled for microglia by IBA1, and we have also PI in magenta channel. It goes through gray matter, a lot of microglia, white matter, few microglia, and gray matter, again, a lot of microglia. These are really hundreds of millions of now cells are visible in one big tissue. Even you can see actually the processes of this microglia. And again, we have large data problem solved by Rami in this work, who developed the algorithm, said now we could, for the first time actually in such a setting, quantify hundreds of million cells using deep learning and really at higher accuracy and speed. So maybe it sounds interesting, but you may not be able to yet have the application in your mind. So why do we need to make human organs transparent? So first, clear applications for us is studying cellular and molecular mechanisms of diseases in an unbiased way. For example, we got a kidney from 92 years old person, post-mortem donation, made it transparent, labeled with some dyes, and look at this kidney at high resolution. You can see here, green shows capillaries throughout the kidney. This is the intact human kidney. And magenta shows the glomeruli. If you see now on the left side, there are a lot of capillaries. On the right-hand side, actually, there is few. And if you look at now magenta channel showing the glomeruli, also we have fewer glomeruli on the right-hand side, while on the left-hand side, we have a lot of them again. So looking at the whole kidney like that, we actually identify previously unknown pathology we can call, because we have been consulting with the kidney experts. None of them could tell us what is this, because they have simply never seen something like this before. So we are definitely very interested in mapping human brain. As you know, human brain mapping is one of the flagship projects of all sciences. billions invested in human brain mapping projects in the EU and the US. Since more than 12 years, there is no single map of a human brain. Well, the reason to me is very clear. This organ is really big, and we don't have optical access to cellular level details. Now, making it transparent using channel technology, we actually open the way. Now we can scan using fluorescent microscopy, potentially the whole brain, and obtain cellular level information, and be really close to mapping the human brain. Finally, we would like to use human organ mapping to generate artificial human organs using 3D bioprinting. So you can imagine that, that when we have the maps of human organs, such as here, human kidney, that will allow us to do the printing, because 3D printing works. Well, 3D printers are 3D copy machines. You have to provide very high detailed 3D map, then it will replicate it one-to-one. That's what they do. If you want to replicate now a human organ, what you would need simply is at least cellular level details of human organs. And now using this technology, we can get them, and we can generate some small organs and large intact whole human organ for transplantation purpose. Finally, to summarize, the whole thing is actually, for me, the concept, as I mentioned, we focus on accelerating discoveries in biosciences to reach to era 2. And that means we have to do unbiased. That means we have to study whole tissue without selecting an organ or a piece or slice. We have to look at the whole molecules without splitting again, whole transcriptome, proteome. And those technologies should be scalable. That means you should be able to do it high throughput. And they should be actually targeted to be most effective in human treatments, that we have personalized and targeted treatments. And now we have on the way to do this by combining technologies from biosciences, AI, nanotechnology, and engineering. So finally, I would like to share with you one of the most favorite videos of mine, the human, not human yet, but mouse brain, this Taiwan GFP mouse, where we see a subset of neurons are labeled with GFP. When we come close to the cortex, you can see all these cortical neurons, their extensions, the dendrites and the branches, all this spacing, enormous amount of details, their connections. And then you go backwards to the deeper brain regions, you can see the cell bodies, but not only cell bodies, where you see all the axons coming out from this individual nerves, cell bodies going to the other side through corpus callosum, in the brain structures. And we have this enormous details in intact mouse brain. Of course, here 1.5 centimeter cube tissue is really amazing. There's about 100 million cells here that could be now imaged in intact setting. Now imagine using this technology, we can quickly and easily catch changes very early on. If it is a mouse model for Alzheimer's disease, now we could see the neuronal changes at very early stages, because we have the whole thing and can easily judge what is changing compared to rest. With that, I want to thank, of course, many funding organizations, and most of this work has been done in my labs at LMU, ISD, and we are now also located at Helmholtz Center, Munich, where our new institute is present. I'm very happy to work with a multinational and multidisciplinary team. We are about 16 countries in 22 people. We have chemists, physicists, computer scientists, of course, biologists, and well, many others, engineers, working together to tackle the questions of that I was talking. With that, I'm at the end and happy to get your questions.
Wow.
Hello.
This is absolutely wonderful. Thank you so much for sharing this research. Also, really 5 years of research that you packed within 30 minutes. So I can imagine that the challenges are very high there. I'm just looking in the Q&A session to see if we have some questions already or not. So I'll start by my own question. So the first thing, when I see all those images, is how challenging technically, at the technical level, how challenging is it to pre-treat the tissue to be able actually to images the way you do it, like with the, yeah.
So overall, how challenging, of course, this may sound a little bit off, and if you haven't done, it might sound very complicated. But I must say, if you're doing histology, have used confocal and so on, compared to that, this is even easier. So transparency, to make tissues transparent, this is a very simple protocol. You have the tissues and put into solution 1, solution 2, solution 3. More or less like this, nothing more. And there are different clearing technologies. Ours are organic solvent based. You mentioned also disco clearing. So these are very easy and very effective. Therefore, potentially they are the most commonly used and widely adopted. And after making tissue transparent, to do imaging, you can, what ideally one needs is so-called light sheet microscopy. This may be less common. At the moment, every major city has at least one you can collaborate, but it's not like confocal, every institute might have one. But having said that, what you can do is also to image the samples using confocal or 2-photon microscopy. You can make some mounts, glass mounts, and then go there and get high resolution, this time whole depth of tissue, which you're limited in confocal. If you do 20-30 micrometer, you will lose the view, but after transparency you can go several millimeters. So those are all easy relatively, and hardest might be the data analysis.
Yeah.
Therefore, that is, of course, when you have so many thousands of images, pictures, what are you going to do? Might be difficult. Therefore, we have been developing deep learning algorithms.
Well, interesting. And I really want to come back to the story that you shared about the connection that you found between the skull and the meninges, right? This is something pretty new. I think there were some guests out there, but nobody had the proof that something like that exists. And like, I like speculation. How would you, because you mentioned that that could be a way to diagnose and treat diseases. And the question is, how would you speculate the route to get to that diagnosis just by looking at the skull?
Yeah. This is actually my ERC project. We got 2 million to investigate this in the next 5 years. So first, I think this is a bit crazy, to have connection from skull to the brain surface. I mean, everyone thinks the skull is like a helmet, right?
Yeah.
Without any direct functional role on the brain, but it's not true. It's totally not true. Something that, people till now believed in simply is not true. There is a functional role. Contribution of skull seems to be to the brain, brain being. So imagine you have these small channels, and in the skull marrow, there are millions of immune cells and their progenitors. Like in our experiments that we did with stroke, we could see them, they get into the brain quickly through these channels. So that means in terms of therapy, one could block this movement from skull marrow to the meninges, right? That's in terms of treatment, these kind of things can be considered. In terms of diagnostics, imagine these gates connections could be both ways. The dirt being generated in the brain might be going out through these channels. We can think about plaques in Alzheimer's, cytokines coming from inflammation in different brain diseases. Maybe they'll go from there into the skull. Maybe monitoring the skull using some new optical imaging technologies, we might catch these brain-specific cytokines, which they should not be out, but when you see them, they're out. You know that something is wrong in the brain. So that's our thinking in terms of diagnostics.
Mm-hmm. Very, very interesting. We have here Taha, who is asking, we're saying, thank you for this great talk. What is the current resolution of brain mapping with Channel? Were you able to resolve dendrites and dendritic spines?
Good point. Well, actually, at this point, if you think about whole human brain, what I showed, we don't have way to image it. So basically, there is no microscope that you can take the human brain and put, but there's no fluorescent microscope. Of course, there's MRI and so on where you don't see the cells. I mean, you can see the cells with fluorescent microscopy, but then there is no microscope because no one made such a microscope because there was no such a sample. We have such a sample. We created a problem. We are trying to solve the problem to make a microscope that can scan the whole human brain. So at this point, of course, we don't have, but when we do centimeter cubes, there we can go to scan voxel size of 6-8 micrometers. That is actually pretty good considering a cell diameter is around 10. So that would allow us to see the cell bodies and potentially dendrites, but will not potentially show us the axons, tiny axons. Some of the axons are a few micron, even smaller. Well, before that, even we have another problem that we cannot label first dendrites and axon easily in human tissue. Of course, mouse, you make transgenic express GFP, fine. But with human tissue, we don't have that. So we need there some new labeling technologies, potentially, of course, post-mortem that we cannot actually give dyes or stuff on living humans. Yet, maybe in future, there's some smart ways to do it, but yet not. So I assume we are in need of some developments there in terms of labeling and microscopy. When we get them, potentially we will have some subcellular level of scan of human brain. Maybe still not the most ideal, like showing every tiny connection, but at the end, I believe we don't really need all tiny details.
Great. Well, in the same direction, Ebenezer Ajayi is asking, can Channel differentiate between health and not so healthy brain? Like with this technology, can you actually discriminate between unhealthy and not healthy brain?
I mean, at this point, as I mentioned, we cannot scan the brains, whole brains, but using the small pieces, yes. For example, we could do microglia labeling throughout really large human brain tissue. And if you have something coming from healthy human brain versus maybe someone with Alzheimer's, we would be able to see the changes in microglia numbers and morphology. And you will need, we are working and doing this kind of experiments now. I think the most exciting thing using this technology is we would be able to tell things at much earlier stages. Or the changes, very tiny scale changes. I mean, a good example might be nowadays corona infection. Maybe in the brain, there's few regions, few cells infected, seems not a lot. How are you going to find them? People are doing histology, but it's just digging, and they don't really find. But now with our technologies, making whole thing transparent, looking at the millions, hundreds of millions of cells at the same time, we can easily catch small, tiny changes, which is not otherwise really doable.
good. Well, this is taking us then to the method that you're using to actually catch those tiny changes. You're managing a lot of data, and Paul Dalton is asking, could you explain how deep learning is used to combine the images into a 3D map? How do you do that?
Actually, we don't really, at this point, use deep learning to combine the images. I mean, after making transparency, we need to tile scan the cubes. And this combination is done using some software, TerraStitcher, Big Stitcher, and RVC Mars they can do. That actually don't really need deep learning because stitching is relatively an easy issue, which potentially could be improved with deep learning. We didn't really focus on that yet. But the major problem is in mapping, then finding the structures. You need to find the cell bodies, the extensions, the microglia. Those are not doable by any technology, any filter, image, RVC. Those are all gonna fail terribly because they are not deep learning based. Thresholding doesn't work when it comes to hundreds of millions. So then we use deep learning, machine learning. What is deep learning? Deep learning, those are machine learning algorithms where you train algorithms like human being, tell them using some data saying, okay, this is a microglia. You see the cell body and processes. If it looks like this, it's a microglia. You show them 1 and 2nd and 3rd, 100, 1000, and then the algorithms learns. Then they don't need to, they know what is a microglia. If you then put whole data in with billions of cells, they will spot quickly what is microglia, what is not, because they learned it. And this learning is done using this usually convolutional neural nets, imitating a little bit of brain networks. There are layers of computational units where information are stored. Going from top to bottom becomes more abstract, and that helps algorithms to be faster and accurate. I mean, you can think this is the same as a human being. Let's say you have a new colleague coming to office. The first time you need to look carefully, how is the size, face shape, the nose, and whatever. But then when you see the guy again, even sometimes you don't need to look. From the shadow, from something quickly you can understand because you know it. Yeah. Exactly. So all the abstract features from this guy, the food, sound, steps, and this algorithms exactly does that. They actually, when it's about image, they use down sampled image, make it more blurry and try to guess it again, make it challenging for themselves. And then doing that, they learn being a cell or microglia from much more abstract information. You don't need the sharpest and clearest image, but they can still recognize because they learn. So this is how deep learning works. It's extremely powerful and definitely will be potentially the only way, I mean, machine learning at least will be the only way to tackle most of the biological data.
Yeah. Well, we're getting some technical questions again. One from Sarwa Lin. Thank you for your talk, she's saying, or he. I would like to ask, what about this clearing method? Does it require high volume of antibody for 3D staining of the tissue, for example? How does it work? Yeah.
Good point. So depends, of course, antibodies. I mean, let's talk about mouse, whole mouse body. Labeling the whole mouse body is crazy if you think actually. We do it using nanobodies. And actually, it's not bad. I think when we buy nanobodies, I don't know, maybe in the wild, there is about 100 microliter, let's say. And I think we use 30, 40 microliter for one mouse. So basically, you can label even 3 mouse with 1 vial of the nanobody. I think this is already crazy. So this looks like it's not really, you don't need liters of things. Even part of a vial, you can label the whole mouse. We will see how it's going to work with the antibodies and whole human organs. Let's say if you want to label whole human kidney, potentially you need some more vials.
Yeah. Yeah. Well, I guess that would be the challenge in a limited research environment, like some researchers in Africa are facing, right? I have actually another question that's more on the transferability of this type of work. So the work you've been doing is mainly ex vivo, so extracting the organ, for example, clearing it, imaging it. But what is the future of this type of method in terms of applicability in the clinic, where you actually want the people to stay alive while you're doing all those things? Are we going to be using contrast agent, for example, to image? How is it going to work when we get to that point?
So, I mean, well, we cannot do transplant human yet.
Yeah.
That would be, I don't know if it will be desirable. Of course, everything we do means dead tissue. As soon as you try to make something transparent, you kill it because there's dehydration, organic solvents, cells, they don't survive that. And what would be the biological application in clinic is, well, biopsies are very good examples. You can take the whole tumor. Instead of pathology doing tiny section and looking at tiny piece of it, we analyze the whole thing. You know about the entire tumor heterogeneity in the patient or in this biopsy. That's, I think, huge advantage. But other than this, what you can do actually, well, you don't need to do this in, I mean, if you just simply think about cancer metastasis, why would you need to make the human transplant to see every single cancer metastasis in human? No. Sounds interesting, but what are you going to do with that? If you cannot treat the person, that has no meaning. So that means actually it's more important to make things transparent at the preclinical stage, where in the mouse, for example, you can develop the treatment. So what we can do is that take out, for example, the tumor from the person and put into mouse. So because now it's personalized, the same tumor from that person. In the mouse body, we can try different antibodies, for example, for the person's cancer. And if we find something from the mouse using this whole mouse transplant, we can bring it to person for treatment. I think that's already really good enough. We don't really need to make the human tissue transparent. It's not going to give us much.
Well, thank you for this answer. Levgeny is asking if this technique can also be used to study internal structures of bones and teeth that have like inorganic compounds. Can those inorganic compounds actually be removed during the clearing steps, for example?
I don't really understand what is the question. I mean, let's answer like this. So during clearing, we use alcohols and organic solvents. Of course, when we make the clearing, alcohols are gone, then the organic solvents, they are there. We do the imaging with the organic solvents because they do refractive index matching, which is a key step. If you use some labels for cells, maybe for bones, I mean, we labeled the cells in the bones. If you label, I don't know what are the other structures in the bones, calcium, stuff like that. So basically, these labels will stay. They will stay because the tissue is in before clearing, it's fixed. Maybe this actually will be more important information to give. So when before clearing, there's PFA fixation, that everything is cross-linked, covalently cross-linked in space. And during clearing, therefore, everything stays in this position. What is removed is the water, dehydration, and also some of the lipids. All the protein structures will be in their exact position. So therefore, you will not lose any information in terms of structure and proteins.
Oh, fantastic. Actually, on a more career perspective expert. So you mentioned at the end that you have a very multidisciplinary team, international as well. And I guess for an early career researcher, the question is, how do you select, how do you choose those highly talented people with whom you work? Do you have a technique and advice on how to select people you want to work with?
Yeah. I mean, of course, this is not an easy to answer, but I will try to give some advice. I think important thing is throughout your studies, your PhD, your career, you have to have a story to tell what you're working on. You know, if your story starts with an MDA 1.2 and structures, people will get lost. Yeah, you have to learn how to present things simpler, general, and make it exciting. You have to develop a good story, and that will be important for your presentations when you go for interviews, when you write a grant application, when you write a paper. Those are all should be a coherent, simple story. Well, of course, no need to say you should be hardworking, excited about the science, and willing. Actually, you should show already you push the limits. You know whatever task is given to you. I'm not. You know, you're not at this point. Nothing we do is rocket science. I would not be just. I'm not interested in someone who's doing teacher transparency. That doesn't help me because I can teach the person in a few days. But if the person doesn't have intrinsic motivation driven for science, let's say, at least for me, would not be very exciting to work with that person. So it should be, people should be excited about science, about what they do, about the field, what they're interested in, and where they want to go. I mean, they should show this excitement, and sometimes you fake it until you learn it. And but it's okay because you may not really know the details before you go, but you say, I'm already excited without knowing. I come, I learn, I will be still excited. You know, you have to give this impression. I'm going to do it. I'm going to learn it. I'm going to push it. That has to be clear. I mean, you have to be, of course, persistent, communicate with people throughout your career, not just during application, but maybe through social media meetings, people you think can be interesting for you to communicate with them. You know, simple things I think can be done. Not many people do. Just write email to people when they publish, say, oh, great paper. I enjoyed it, you know. This is, I think, a really good way to get some attention because simply no one really does that. And even if the top leader in the world, Nobel Prize laureate, receives such an email, you will be noticed because potentially no one really writes to him either, right? You, through Twitter, through emails, you can show yourself. I think that's also important. Also have a mentor. Always check someone can help you. You can write to people even you don't know them. So, okay, I'm XYZ person. Can, you know, briefly describe yourself and you're interested in mentorship, getting mentorship, how to be a successful scientist like that person. And then you have to be very clear also with what you need, like, you know, saying, okay, I would like to meet you once in a year or twice, a few hours, 1 hour. I will have clear questions about how to move to next level, about my career, what you will advise, you know, these kind of things. Very concrete, clear, to be clear from the beginning. And then people who would be, again, I think, willing to accept something like that. Again, many people don't ask. So that's surprising to me as well. People don't look for free advice. They look for advice when they come, like, you know, I don't know, a few weeks, a few months before the end. Well, that is actually a little bit of they look for an escape to that. Usually it's in such position, people would be less willing to help, because if you're desperate, people are not very interested in. But if you show your interest from the beginning, you did, just because you're interested in, it's always more attractive. So those are some suggestions for the career, I guess.
Well, thank you for this career advice. You really, or mentorship is a big issue, again, especially for the early career researchers who are not privileged to have all the instrumentation and so on. Sometimes they're just lost and don't know what to go or what to reach to. So I guess you will be available if we have some of our participants sending you an email and saying, hey, that I took, like we have now, we have one participant who said, I'm happy to listen to you here again after having first listened to you on the HCA webinar platform. This is coming from Ebenezer Ajayi. So, well, thank you for giving us your time today. Thank you for answering all those questions. This was very insightful. And yeah, I think we'll close the session here.
Sure, great.